So my name's Jim, and you are here because we have a workshop, and the workshop is the NGS Hadoop workshop. So this is, I think it's almost a historic occasion because I haven't been involved in this area very long in terms of NGS or bioinformatics. I'm not a bioinformatician, I'm a distributed systems person. But there hasn't really been any official workshops or meetups where people who work with NGS data, bioinformaticians, talk about Hadoop, exclusively about big data. So we're calling it Hadoop because Hadoop is the dominant paradigm in big data. So we tried to bring together a bunch of different people from all over the world, in fact, who are involved in building data-intensive computing solutions for NGS data. And I think we've got a really great lineup today of speakers. And tomorrow, for those of you who are practically inclined, we're going to be doing workshops, tutorials, and hackathon. So I looked at the line of people coming here. I think about 23 out of 60 people are from outside Sweden. So quick guide to Sweden, for those who don't know it. Spotify, it's pretty cool. H&M. We have a lot of big companies. We have Nobel Prize. Maybe you're talking to your auntie before you said, I'm going to Sweden to give a talk, and they thought, okay, maybe you're going to give a Nobel talk, but that's somehow aunties or something. They don't really know much about our area. The one key word we have is fika. You hear it everywhere in Sweden. It means basically just to drink coffee, have a bun, and we'll be doing a bit of that today and tomorrow. So the other thing you always hear when you come to Stockholm is sometimes people pronounce it archipelago. It's archipelago. For those of you Swedes who pronounce archipelago, it's pronounced archipelago. What is it, right? Because people always talk about it. Stockholm has this fantastic resource. It's actually all the islands, the thousands of islands outside Stockholm. It's one of the largest natural archipelagos in the world. So when people talk about it, you know what they mean when they say archipelago. Today you're at KTH. It's a university. It's a technical university, to be more precise. It's quite old, founded in 1827, and it's quite large. So it's one of Europe's largest technical universities. You're now in Kista, which is a suburb of Stockholm, which is like 15 kilometers from Stockholm. The main campus is downtown. This campus is here primarily to serve the large companies such as Ericsson that you'll find here in Kista. So in Kista, every day 20, 30,000 people come in to work, and every day they go back home to where they live, and not that many live here. So it's kind of a working town. So some practical matters. We'll have lunch. It'll be on the second floor. I hope we'll just go there together. You'll see the different stairwells. One of them's marked C. So when you go out here, it'll be on the right. And we're going down a floor. So it's on the floor below this floor. Coffee we'll have after lunch just outside here. And tomorrow, we won't be in here tomorrow. We're going to be on the top floor of this building. So there's a 6th floor. Actually, there is a 7th floor somewhere, but I know it's a magical floor. But most of the floors finish at the 6th floor, and it's an organization called SIX. They're a research institute. So we'll be in a room called Knut there. And the workshop, we got, thanks to some sponsors that are behind this. We have the Swedish Science Research Center. We got a bit of money off the KTH Big Data Platform, and then we have some projects behind this, such as End-to-End Clouds and BiobankCloud, an EU-funded project. So I'm aware that many people here are not distributed systems people like I am. I think is probably 2/3 are bioinformaticians as far as I can make out, and 1/3 are distributed systems people with an interest in bioinformatics. So I thought I'd just do a quick description of Hadoop, because we're going to have a lot of talks about people's particular platforms and solutions, and maybe we need a little bit of background knowledge to fill in things. So if we go back to, let's say, 2004, when Yahoo were building search indexes every day. They were downloading a lot of data every day with crawlers, and they were building a search index. The kind of infrastructure they were using is one which we would say has no data locality. What you would have is you'd have a bunch of grid filers or large storage-intensive machines storing all the data. And at nighttime, when you want to submit your job, which may be building the search index, if we're doing genomics, it may be processing genomes. What would happen is you would submit your jobs to compute grids. You would have maybe separate blade servers that would do the computation. So it sounds like a good architecture. You have nodes specialized for storage, nodes specialized for compute, and you send your job to your compute node. The thing is that it needs to read up all the data over the network to get from the storage nodes to the compute nodes. Now, what Yahoo found around 2004 was that the amount of data they were downloading every day was more than the amount their network could transfer over to the compute nodes at nighttime. They literally couldn't keep up with the architecture they had. So the problem here is that this doesn't scale. Bandwidth is the bottleneck. I'm talking to you HPC people who are here. Okay. So a lot of genomics people are aligned with HPC. HPC, I don't know if they've, what happened to my slides here? Oh, we lost contact. There we go. So a lot of HPC doesn't build on this principle because they're very compute-intensive jobs. But for I/O-intensive jobs, this model doesn't scale. So what is Hadoop? And there are many different frameworks. There's more modern in-memory processing frameworks like Apache Flink and Spark. But MapReduce is the classic one. So just to give you an idea of the execution framework in MapReduce, what does it mean? So the difference here between the non-data locality implementation here, where the data was read over the network, to the compute nodes where the computation was performed. In this case, we'll send the job, the computation, to where the data resides. And that's the principle we call data locality. So what happens is you submit a job in MapReduce to something called the job tracker, and it will then say, OK, I'm going to process this file here, this genomics file, this BAM file. And the BAM file happens to be split up over all these machines. And you can see we have numbers, 1, 2, 3, 4, 5, 6. They're blocks that make up this file. And we can see that the blocks are split up over all these different machines. So what that means is that I can now execute this job in parallel. You can submit the job to run on parts of the file in parallel. So we submit our job down here. They're going to read the data locally from disk, and they're going to execute on that part of the file. And they'll write the results maybe to local file, maybe to a distributed file system. But there'll be much less traffic over the network. So in this case, the network bottleneck won't be as big a problem if we're not shuffling vast amounts of data across the network, which you shouldn't be if you're doing writing a proper MapReduce job. OK, so that's the basic principle. So data locality is what differentiates big data and Hadoop solutions from previous solutions. Now, the thing about this is, if we're thinking BAM files, and you're saying you're a bit smarter than I am, and you said, well, hang on, the BAM file, the header file is on this block, but how can I do some computing in this node? There's no header file for the BAM file on this node, or this node, or this node. And that's true, right? So the problem is that, well, maybe our formats are broken. That's one thing, like BAM format for a big data model. And the other thing is that the computations that we wrote previously don't map naturally to this model, because we have to perform computations on small parts of the data in this model. So the MapReduce programming model is slightly different. And an example I give to define this is, you can imagine a vote counting machine. So, you know, if we think of the election we had in Sweden last year, you have lots of vote counting centers over the whole of Sweden. And we don't send all our votes to one single place like you would do in the grid computing example, where you upload all the votes to a single place where you do the counting. No, you can do the vote counting at the local voting centers. And then you just need to send the statistics up. So it'll be much less data transferred up to the central place where the votes are all counted. So the MapReduce programming model involves scanning your data. And you scan your votes, and the votes will be filter out the ineligible votes, and sort them. So each candidate would get a bunch of votes that would end up in each of these cubby holes. And then we can do some reducing to count the number of votes for each candidate, and then we can send off the results to the next phase, which counts the count of counts and so on. So you can chain MapReduce jobs in that way. So the difference between modern MapReduce programming and these batch sequential processing machines is that we have fault tolerance. So we have thousands of machines, and if a machine fails or crashes, the computation continues. That's it. OK, so that's the basic introduction to Hadoop to begin with. And now a little bit on why Hadoop is interesting for genomics. So in genomics, many of you will be tired of seeing this slide. If you're working as a bioinformatician, the cost of sequencing whole genomes is dropping dramatically. It's about $1,300 now. And it's dropping much faster than Moore's law. It has been doing so for a number of years. But the reason why it's dropping is because we're getting new sequencing machines that are producing lots of data. So I prefer this slide, which tells me, well, if I bought the latest Illumina HiSec X10, how much data will it produce per year? So it's going to produce a volume of 5.2 petabytes per year. And the velocity, if I have this machine switched on all the time, which I should do, because why would I buy it otherwise? It'll produce about 45 megabytes per second. Now, I know that SciLife Lab here in Stockholm and Uppsala bought one. They split it up between the two of them. And now they have to transfer this volume of data per second, or maybe half of that from Stockholm to Uppsala every day. and then there's issues with that, right? Getting that over a single TCP connection is not a trivial matter. So these new machines, next generation sequencing machines, are producing big volumes of data. The thing is that a lot of the variant calling pipelines we have today, what they do is they'll take the data from their big NGS machine and they'll send the raw genomic data to a machine, and that will process it locally. And okay, we get some parallelism out of this, but the problem is that each of these machines is going to take roughly 24 to 36 hours to process all of this. It's not going very fast. We're not splitting up the data over lots of machines and processing it in parallel. And then these genomics pipelines that we have today, they're reading and writing lots of data to disk. They're I/O bound. They typically run on a single node. So the question in this workshop what we're dealing with is, can these genomic algorithms that we have today, can they be reformulated or rewritten as data parallel or graph parallel algorithms? Hopefully you'll get some answers from many of the speakers that we have today. And what we'll talk about as well, what we won't talk about too much is there are many platforms out there for genomics that are appearing every day. So this year we saw Google, or last year we saw Google Genomics appear. Illumina have BaseSpace. You have the Beijing Genomics Institute, 23andMe. There's a lot of commercial activity happening in it. But the question for us, I guess, for many of us are interested in, are what are the open source alternatives? So what other platforms are here today? So we have, like I said, a number of speakers today. What the speakers all have in common is that they've all built systems based on Hadoop or some data processing framework that runs on Hadoop for NGS data. So without further ado, we'll take the first speaker, which is me. And I'll talk, I think I'm almost on time. And I'm going to continue talking here. So the other speakers we have today, not everyone's here at the moment, but they're from pretty much all over the world, in fact. So we have from Berkeley to Germany, Belgium, Italy, Finland, and Sweden. How's this going? It's thinking about it. There we go. Okay, so I'm going to talk now about BiobankCloud, which is an EU-funded project. It's a European project. We have a number of partners from Karolinska, KTH here in Stockholm, Charité and Humboldt in Berlin, and Lisbon in Portugal. I'm going to talk a bit about population scale genomics and how we can help realize that vision of population scale genomics with BiobankCloud. So to start out with, one thing that's very important, we're gone again. There we go. Sample sizes matter. So in Europe, we have quite restrictive legislation about where biological samples can be stored and where the data for them can be stored. So it's not obviously trivial to upload all your samples to Amazon. Often you're prevented doing so by legislation. But the larger the number of samples you have, the more statistical power you have, the more inferencing, the better inferences you can make, or more inferences you can make in your data. And here's a really good example. I don't know if this one is as widely known, but the company 23andMe, who have a very large database of not whole genome sequences, but they have their own genotyping kits, which do, I think, 200 to 300 genes they sequence. But they looked at their data, and they saw this thing, these outliers here. So they were looking at this particular chromosome. You can see here at the gene that's nearby. And they saw these outliers, and they said, okay, they had so much data, they saw the outliers. If you didn't have that much data, the outliers might look like this, and you're not sure if that's statistically significant or not. So the line of statistical significance probably lies somewhere here. Okay, so the red ones are within, see the red dots there, they're statistically significant. So then they said, okay, let's get some, have we any hypothesis of what the implications of these mutations could be? And they sent out a number of questionnaires. And I'm not sure exactly how they got to the questionnaire, but they found out that these mutations are highly correlated with people who have a propensity to sneeze when they go in light. So when you go in a bright sunlight, you open the door and you sneeze, right? You have a higher probability of having one of these mutations. It's called the photic sneeze reflex. That's kind of cool. So the bigger the sample size you have, the more empathy. I don't know if that was part of the question. Is that part of the questionnaire? I don't know. As I said, I don't know how they actually came along with this. Maybe they just had a field to say, do you have X, Y, Z? Obviously, there's implications for how big your questionnaires should be. The bigger they are, the more you can pick up. But then you have people haven't got infinite amount of time to answer these things. So bigger data sets give you stronger statistical inferencing power. This thing is acting up on me now. So what you can say is that at the moment in the Hadoop world, we kind of live in this centralized data set world. I talked to biobank people, and they really want to go with federated data sets. We don't have the technology with which to do analytics over federated data sets at the moment. I'll try, that's a good idea. We actually have a left one as well. Has anyone got a VGA to Mac adapter or something? I think I might have one in here. All right, great. Let's try that one out. Thanks. See if this works. There we go. Okay. Yes. Okay, so I was talking about the current solutions we have. So Hadoop gives us the ability to build large-scale centralized data sets and to perform analytics over those data sets. So they're technically feasible at the moment. Politically in Europe, we have issues with collecting large samples, amounts of different data sets and putting them centrally because, like I said, we have often laws preventing us from doing that. So a lot of people think federated data sets are the way forward, which are politically feasible, but technically we don't have the technology to deal with them. The goal is really to have more samples. So the more samples you have, the more power you'll have to make insights into disease and the genetics behind disease. So today I'm going to talk mostly about centralized data sets and BiobankCloud in that. So here's another example of why we need faster whole genome sequencing pipelines. In the UK, there was a bacterial outbreak in Birmingham last year, and they needed to sequence the bacteria if they found a novel strain of a bacteria, A. baumannii. And without fast sequencing, it would have taken them a longer time to identify the bacteria and halt the outbreak later. So the faster they could sequence it, the faster they could identify the cause and then stop the outbreak. So, you know, at the moment, if we're looking at 36 hours to go from raw genomic data and FASTQ files to getting our BAM files and variant files, that's quite a long time. You know, the goal, I think, some people will talk later on, we'll talk about how we're going to get this down to a couple of hours in the near future. The reason why the serial pipelines are not very good is very simple to explain. If I want to read a whole genome from my hard disk, and I'm working on standard commodity hardware here, what we're looking at is really 1,000 seconds to read up a 35 times coverage BAM file. That's pretty slow. Now, if I split that up over 1,000 machines, imagine I split my genome up over 1,000 machines, and that's we're putting blocks of our file across, striping them across many machines. I can read up my whole genome in a second, in principle. And that's the basic principle of data locality that we were talking about earlier on. Another thing, again, this is just repeating the point about Hadoop is not exactly the same as HPC. Hadoop and big data runs on commodity hardware. So you buy cheap boxes. This is a pretty extreme example, but you can buy boxes that are intensely very disk intensive. This example is obviously very little compute, but it's quite cheap. If you really wanted to, with these Backblaze boxes, you could build a cluster that would store 133,000 genomes for just over €1 million, which is pretty incredible. Now, you know, you're not going to use triple replication in HDFS. We have something called erasure coding, which will get down your storage costs. But The principle is that you can do very large-scale, population-based scale genomics on commodity hardware, and the cost is reasonable. So the cost, this would, handle my Illumina X10 for the next, 4 years or whatever, all the data for it. So I don't know what people typically have as budgets for compute infrastructure when they buy Illumina X10s, but I know that not everyone actually thinks in those terms, and they budget appropriately for buying the compute infrastructure. So what type of networks does this commodity hardware run across? Well, typically you might have either 1 gigabit or 10 gigabit switches at the top of your rack, and then they'll be connected across by either 10 or 40 gigabit links across spine routers. And one of the interesting points is you'll always have oversubscription. What that means is that the capacity on these links will typically become the bottleneck in your system, because they don't have relatively as much capacity as the links between the nodes. So what Hadoop tries to do is implement things like block replacement or block placement policies so that you minimize the traffic across the inter-rack switches. Hadoop itself can mean low administration costs. So if you're running a cluster and you're allowing people install whatever software they want, maybe with the help of VMs, or maybe even not even with VMs, or maybe on bare metal, you have a lot of administration overhead because people can do funny things and break things in unusual ways. Facebook run Hadoop, and in northern Sweden they have a massive data center in a place called Luleå. I think they have like 100 or 200,000 machines, I think. And for those 200,000 machines, roughly, they have, I read somewhere 33 employees, right? And one of the VPs of Facebook said that each Facebook ops staffer manages up to 26,000 machines each. So, if you have 33 people working there, you get 8 of them on call at a time. They manage your entire cluster, or the data center, sorry. And, that's not a huge amount of administration overhead. Now, okay, if things break, you maybe fly people in to fix them. But in the general case, it's not necessarily high administration costs of running large Hadoop clusters. Okay, so that's a bit of background. So what is BiobankCloud? So one thing we have an issue with, for bioinformaticians don't always know what biobanks are, but a biobank is effectively a place that you store human material and human biological material, and you can identify the human associated with it. In Sweden, we have a number of projects that are starting up large biobanks. In Finland, they have very interesting national biobank legislation where all the hospitals collect samples to central places. We have a project called LifeGene, which the goal of it was to store biological material for 500,000 people. And what we want to do eventually is sequence this data, right? So to do even whole genome sequencing on all the data. So the BiobankCloud kind of comes from, well, what happens if we sequence the data in biobanks? Who will own that data? I mean, it'll be the biobankers who own it and manage it. And you who are bioinformaticians and want to do analysis on it, ultimately they're the people who will be providing you with the data. So biobank we have a number of different actors in BiobankCloud who are interested in taking sequence data from biobanks and doing things like storing it securely, doing analysis on it. You have IT admins who manage clusters. You have ethics people and auditors, biobankers and bioinformaticians. And we need them all to feed in together into the same ecosystem. So one of the issues is that Hadoop, as it currently stands, is not, it doesn't have all the tools that we need, right? So one of the basic points in Hadoop that's a problem is that you need multi-tenancy. So if you have study data, clinical study data, and somebody else's clinical study data, can you keep it on the same cluster? So I know that here in Sweden, at one of the large sequencing centers, they have separate clusters for production sequencing and also for research, because they're worried about people accessing data that they don't have authority to access. So Hadoop itself doesn't give any straight out of the box support for multi-tenancy for studies. There is low-level authorization support in the file system, and there's some projects to do centralized management of audit logs like Ranger, and then there's projects to do authorization at the table level in databases such as in Hive or REST APIs in Knox. But these aren't enough, right? There's not enough in that. The other big problem we have in Hadoop for NGS data is that the formats that we have in NGS data, like the BAM format, it has a header at the top. And when we break up our file and put it over lots of different nodes, the blocks that header file won't be on all the nodes. So we need new formats. And today we'll hear about a couple of other examples, like Hadoop BAM is one such format. So the other issue is no standard API for querying genomic data. So Google have introduced recently Google Genomics, and that you can go in, log in, and look at their SQL-style API for querying genomic data. You know, there's a couple of proposals for standards, but there's nothing really that's you can say is dominant at the moment. Hadoop itself is scalable, but it's not scalable to the levels we would need for LifeGene project. And Hadoop itself is not trivial. So when we talk to biobankers, installing it, how do I manage our metadata, make it consistent with the data in the file system is not a trivial undertaking for biobank people. So one thing we try and do in the project is we try to bring together biobankers and bioinformaticians. Now, I thought these were the same people a couple of years ago. I couldn't say, what's the difference? Come on, these people hang out together. They don't typically. I mean, biobankers nowadays are starting to produce NGS data, so they're the producers of it. They manage collections and samples. They're typically not programmers, whereas bioinformaticians are typically programmers who do analysis of the data. And what we're doing is trying to bring them together in the same platform. So our platform has a notion of a LIMS, I'll talk about in a minute. And then we have the typical Hadoop stack. We have HDFS and Yarn, and we have our own data analytics platform called Cuneiform, which runs on a schedule called Highway. We also have support for sharing of data. I'm not going to talk too much about between biobanks. It's currently not in our prototype, but we're working on integrating it. And then finally, we have support for installing this as a platform as a service using something called Caramel and Chef. So for sharing of data, we have an architecture called Charon. You can read up on it. There's a paper out there. It's a way of sharing data across clouds and also using public clouds. It's effectively, you can say it as being a distributed file system. But I'll talk a little bit more now about this platform we have for allowing biobankers and bioinformaticians working together. We call it Laboratory Information Management System, or LIMS. But basically what it is an entry point into Hadoop. So typically Hadoop has services. You have services like the file system, Hadoop file system. You have MapReduce platform. You have Spark, Flink, and so on. But what we're doing is we're controlling access. So we're providing perimeter security to Hadoop. So you can only get into Hadoop or our platform through our LIMS. So what we've introduced that's novel here is that we have our own model for access control, and we do it at the project level or study level. So within a study, you have researchers and you have owners of studies, and they have different privilege levels. And we have that as a central concept in the platform, implemented with using the Hadoop Trusted Proxy model. And then on the platform, we also have support for different Yarn applications. So at the moment, we have support for Cuneiform, and then early support for platforms like Spark and Atom and Flink. And there's REST APIs for this, so we can be extended. So what does it look like? So firstly, we have a two-factor authentication for logging in. That means you have to log in with a YubiKey or a mobile phone as well. And the platform itself looks a bit like Git, for those who are familiar with Git. So we kind of call it Git for big data. So when you log in, what you see is an activity trail here, and you see a number of studies. So the studies anybody can create. You can go in and create a new study, and you can invite people into your studies. So you'll have your personal studies that you own, and then you'll also have studies that you've joined, you've been invited into. And then we have a global activity trail for events that you can see, an audit log as such of the system. When you're in a project, you can, or a study, you can invite people in. You can add new members. You can change their, from master to researcher to auditor, change their role in the study. And then we have a file browser. And this basically is a file browser for Hadoop. So you can see a bunch of files here. And this is the root project name. That basically maps onto a folder in HDFS. And everything under that folder is part of your project. So the privileges that you apply or the roles that people have in the project, they get for all subdirectories from there. And different studies can't access directly each other's data that way. And there's some other funky things you need to do in the background in terms of usernames need to also be appended with project names to make sure that if I'm in two different studies, I can't run a MapReduce job across both studies if I don't have authorization to do that. But in general, the principle is that users within a study have a role, and then they can do certain things. They can, if you're a master, you're allowed to upload data and create folders. If you're not a master, you can't. You can just run analytics jobs. So we also have support for biobank, kind of typical applications, managing samples and collections and studies. And then there's an analytics platform called Cuneiform that Ulf Lees will talk about next. One issue we had with the platform is uploading sample data. It's a limitation of your browser. Your browsers don't allow you to upload more than 3 gigabytes, so we had to implement a JavaScript library to chunk files and load them up one chunk at a time, and then do resumable uploading in case there's a problem while you're uploading. For automated ingestion of data, you can use Apache Flume to get the data in here. But they're basically the two ways we get data into our platform. Either users upload it, or we have a production job which runs via Flume. Effectively, what you can see of this is, from a user's perspective, I have this file browser. I have all my samples and my analytics jobs and so on in here. But underneath it, there's, you can have petabytes of data. From a user's perspective, you don't see the petabytes of data. You just see this file browser at the top level. So what we're doing, which is slightly different in the background here, is that we're running on our own distribution of Hadoop, and it's called Hops Hadoop. And what's different between Hops Hadoop and standard Apache Hadoop, or any of the common distros like Cloudera or Hortonworks, is that you can see here we have multiple name nodes, and we also have multiple resource managers in Yarn. So there is a high availability model of the file system, the Hadoop file system, Hadoop, implemented in the Apache variant, but it doesn't give you scale out. So you can't add name nodes to make it scale better, because all of the metadata is stored on the heap of a single JVM in their model, both for the resource manager and the name node. In our model, we took all the metadata and we put it into a distributed in-memory open source database called NDB cluster. And now we can scale out quite a lot. So we can scale out at many different levels. There's internal protocols in HDFS, block reporting, which we can scale out and load balance them over name nodes. In Yarn, there's a resource tracker service where the node managers heartbeat to the resource managers, and we can scale that out as well. And then for the file system, clients can send their requests to any of these name nodes. There is a leader name node to handle coordination of replication events. And we also have a single node to run the scheduler at the moment in Yarn. But we're on our way towards fully decentralizing that as well. OK, so the file system looks like this. You have a client request are load balanced over the name nodes. We have one leader, so we have a leader election algorithm. We're not using Zookeeper, because we have the database. We implement our own leader election algorithm and database. The cool thing here is that we have so much metadata that we can customize it. So some of the things we've done are added support for like block level indexing, snapshots, and erasure coding replication. And that's easy to do because you're just adding tables to your database. You don't have to go into these highly engineered, very tight data structures that's in the existing name node in Hadoop. So what we also have is fast failover time, because these name nodes are pretty much stateless. So in HDFS, Apache HDFS, it takes Spotify about a minute for their name node to fail over if the active name node fails. For us, it takes 5 seconds roughly. Same is true for Yarn. And this is where I guess we kind of, so the performance of this, I mean, I don't have any figures for the performance, but we are faster in writes and reads when we add like 5 or 6 name nodes than existing Apache HDFS. But what's really cool is that we can have much more metadata. So, Apache Hadoop has kind of a limit of 100 gigabytes on your heap, and then you really need to start taking care of garbage collection policies and so on. You're not really going to get more than 150 gigabyte heap size on a standard JVM. There are some specialized JVMs, but typically they're not used. So Oracle's JVM or OpenJDK, 150 gigs is kind of where we're at. And this isn't getting better, so the software technology for doing garbage collection is not improving with time, whereas memory costs are decreasing, and we can go up to terabytes in terms of the size of our metadata. So one thing we did last year, which is really cool, is we added, it's not in kernel Apache HDFS, is we've added support for what's called erasure coding replication. So if you store your data in existing HDFS, and you're, if we take the Illumina X10, if it's spitting out 1.8 petabytes per year, your 1.8 petabytes will become 5.4 petabytes in HDFS. So you're going to store 3 copies of all the blocks. It's called triple replication. So what we did is we added support for one that reduces this to 2.2 copies of the data, which is called XOR replication. This is very nice as a general solution, because if a machine fails that has this block, well, then the other machine can continue running. In this particular running, we have 2 copies left, but in this, if one fails, we can still recover. If both of these fail, we can still recover by using our XORed copies. Now, if we get 4 of these 2 fail and these 2 fail, then we're kind of stuck. But erasure-coded replication looks slightly different. You get only 1 copy of all the blocks, and then you have a number of error correction blocks, or erasure-coded blocks, and any combination of 10 of these 14 blocks can be used to recover an existing block. Okay, so if we imagine one of these has failed, then any 10 that are remaining can be used to recover that block. That obviously means if you have failure, you get a lot of data read over the network. So that's the downside of erasure coding replication. Okay, platform as a service support. So one thing just for a bit of full disclosure, I used to work at MySQL where we developed this database, NDB, years ago. So it's really cool. Like the throughput is 70, 80 million reads per second, transactional reads per second, 100-byte operations. They have insane throughput levels, but it's pretty much unused outside of the telecoms industry, and some other eBay and some other people use it. It doesn't have high visibility, and the real problem with that is that it's very hard to install, right? So from day one, we had this goal that we want to make it easy to install. Now, if you look at Cloudera, which is one of the main distributions along with Hortonworks, and you want to install it on EC2, these are pretty much the instructions you get. So they have a web interface called Hue, and first you need to go into Amazon, log in, create your account, and download your key pair, create key pairs, download data. It's quite complex. For biobankers, this is kind of challenging, let's say. We have support with a system called Caramel. I don't know, how's my time doing? I think I'm okay for time. So I'll just do a quick demo, basically how it runs. You download the app. I don't know if you can see this. No, you can't, actually. Let me just change. I need to exit full screen mode so we can see this. Okay. So what I did was I double-clicked on this executable, and it loads up a page. And from here, you can basically just open up a cluster. So you can have predefined clusters. Here's one for BiobankCloud running on Apache HDFS. It'll run on Apache HDFS as well as our own distribution. And that creates your cluster. So each one of these defines a stack, a set of software services to install. So you have 3 stacks here, and you have 1 instance of this particular machine, and 2. So you can change this to be 100 if you want. So you get 100 data nodes will be created. And at the moment, we have EC2 and bare metal support, but basically you just put in your Amazon key pair, validate it. This is going across the network. Hope the network's working. It is, yeah. Okay, it's a live demo. It's always risky. And then you put in your key pairs, and I have one already, so that's fine. It can generate it as well. And then you just launch it. And then, if you go into EC2, we can see that the VMs will start being created. What the platform looks like if we log in is you get something up like this. And let's reload the page, see if it works. And when you log in, you basically get you can create studies here, and you can go into a study. Yeah, and then you can add people to your team, browse your file system, and so on. And then we have ways of launching jobs and so on. So that's basically the platform. And it's really important for us that this is easy to install and set up, because, like I said, this is, for adoption, it's crucial. So just to conclude, so I've been talking about this BiobankCloud platform that we're working on in this project, this EU finance project, and what it really is trying to show is that open source commodity hardware open source software running on commodity hardware is a feasible way of managing NGS data. So not just managing the data securely, but also being able to perform analytics on the data. What we're adding in BiobankCloud to the existing Hadoop platform is support for our own security model and more scalable analytics. And this stuff, you can, if you want, you can go to caramel.io to test out, early alpha stuff. But we'll be doing a proper beta release this quarter. So this is a team. Thanks. There's quite a lot of people involved in it. And that's it for now. Thank you. Questions?
Header file problem.
Yeah.
If you distribute it, the header file is not on one node, but how do you process it? How did you solve that problem?
So this is not something we've personally solved. This is a community effort, so there's lots of different people working on it. There is a format, like I said, called Hadoop BAM that Kay will talk about, I guess, in 2 talks' time. That's one potential solution. But I would say there's, and that's great work. But the formats that we're going to use to store NGS data, they're not, you can't say that that will be the only format that will be there. There's other considerations, like, so for example, maybe someone will talk about Parquet later on, and Parquet will come in a new version that supports indexing of the data that we don't currently have. So there's lots of technology that needs to be developed. So, we have support for getting down to database level. We have support for projections across columns. We don't have support for indexing rows. And, some of these things will need to be able to do efficient querying of genomes. So I would say that, that's an open research problem still at some level.
And that is the heap issue of the Hadoop, where you cannot store much of the data in a heap, and then you mentioned something distributed in memory cluster. Yeah,
we push the data. Sorry?
What is the name of that?
That's called hops. You can go to hops.io if you want to have a look. But it's, so the heap, so in the current Hadoop platforms, the metadata for managing both the resource framework called Yarn and then the file system called HDFS, they store their metadata on the heap of a single JVM. Now, it is highly available in that they do have an eventually consistent replication algorithm for keeping that with active standby replication. But it's not scale out, so you can't make it bigger, right? You can't make your metadata bigger. There's a limit on the number of files you can store. There's a limit to how much metadata you can store about each file. So what we've done is taken that data and moved it from the heap of the JVM and put it into a database. And there were a lot of challenges there in terms of maintaining the consistency of the data, ensuring it's deadlock free and so on. And then performance is the big one, So doing that in a performant manner.
Is it available online, the open source? Do you release the code? Or?
Like I say, Q1 proper full release, but I can give you access if you want. One more question.
So when you first described the platform as a service, I was imagining it was going to be like a service that other people come and use in a hosted system as opposed to needing to install it themselves. I think you described it as something, a platform you would set up, a local group could set up themselves.
Yes.
Is that my...
Yeah, actually, originally we call it platform as a service in day one. We kind of saw that we need some software as a service as well. Right, so this LIMS application is software as a service, but we're building on a platform called Chef, which is a configuration management. So you may have heard of virtual, everyone knows about virtual machines at Amazon and so on. But how do you maintain software on virtual machines or on bare metal machines? Right, there are platforms like Chef and Puppet that are widely used to do that. They take a bit more time to install the software than something like Docker or a pre-baked AMI, an Amazon machine instance. But what they do is they allow you to update that software over time. So we're building on this platform called Chef. We have written a lot of code to do the automated installation of the software, and it'll run on if you have bare metal machines with no virtualization, it'll run fine there. If you're running Amazon EC2, we've support for that specifically. So yeah, basically any group can do this. The whole idea for us is that we want institutions who have data where the data can't leave the institution, that they can install this locally. So this is for local clouds. There are already very good solutions as well, like I mentioned earlier on public clouds like Google Genomics and so on. Our platform will work on public cloud, but we're really focusing on private clouds or bare metal clusters.
Are there any other groups outside of KTH using it now?
Well, we have the consortium partners, 5 of us. I mean, we've been talking to a lot of people. We have a small project with validating it with SciLife Lab this year. They have a large NGS sequencing machine. So yeah, we're interested in finding new users to test it, of course. Okay, thanks. Then we'll take the next speaker. It's Professor Ulf